Hello friends, Ace Botane next episode. In this episode, we'll be discussing molecular basis of inheritance, 7th one. And what all things we'll be learning in this episode. For that, I will share the screen so it will be convenient for you. I think screen came, so we can proceed. Here in this episode, we'll be discussing as usual, the introduction will be there, then genetic code, transfer RNA, and 5 theory question, 5 NEET model question with the key. So we can enter into molecular basis of inheritance, 7th one. So as an introduction, see in this episode, how DNA carrying message further that will be discussed. And main aspect will be genetic code from the tRNA, structure, nature, functions, different models, etc. So we can proceed to next area, genetic code. Its basic aspects we can discuss. Term genetic code is concerned with a famous scientist, George Gamow. And what is the meaning of the term genetic code? What is the aim? How it works? All we can discuss here. We already know DNA is carrying all genetic informations. And in DNA, only 4 types of bases will be available. Adenine, guanine, thymine, cytosine. So the informations are expressed in the form of proteins via amino acid through RNA. Already we studied transcription. So DNA to RNA to proteins. So final result will be proteins. So it is through an event. So protein synthesis, our next topic, will be through an event called translation. But in all this case, the main issue is polymer of nucleotides in DNA to be connected with the synthesis of polymer of amino acid. So DNA to amino acid, and that will lead to protein. But interestingly, DNA and proteins have no complementarity directly. So this connection has to be done. And that connection is done by the genetic code. I think now idea clear, what is genetic code, what is importance, what is the use. And what is genetic code? Linking DNA with amino acid, okay. Beyond that, it is mRNA sequence, which contain coded information for each amino acid, which contains 3 nucleotides. So coded information respective for each amino acid. When we say each amino acid, that will be represented by or coded by 3 nucleotides or 3 bases, what we call codon. I think that idea is clear to all of you, purpose, aim, etc. Now, genetic code not suddenly developed. It is an evolutionary event, and many scientists involved, different areas involved, different branches involved. And we can start with NCERT, 4 scientists named, that is a usual question for theory, and NEET exam, mass of following contribution, comparison, all this. So we can start with the famous scientist, George Gamow. So our NCERT. So a physicist who argued that since there are only 4 bases, if they have to code for 20 amino acids, 20 here means natural occurring amino acid, the code should be constitute a combination of base. So he suggested for coding 20 amino acids, naturally occurring, the code should be made up of 3 nucleotide sequence or 3 bases together. How? Suppose 1 base equals 1 amino acid. Maximum 4 bases connected with RNA, UCAG. So 4 amino acid, 20 amino acid we have. If 2 bases in combination, then 4 square amino acid, 4 into 4, that is 16 amino acid only, still 4 lacking. If the bases in 3-letter combination, then 4 cube, 4 into 4 into 4, 64 amino acids can be coded. And maximum types of amino acid natural occurring is 20. So 64 means much higher than the required 20. This is how George Gamow explains it. I think idea is clear, direct NCERT. Now comes second scientist, Marshall W. Nirenberg and Heinrich Matthaei, used a synthetic polyuracil RNA and deciphered the code by translating this as polyphenylalanine. So what is meant by the term decipher? Different area it comes. Deciphered means basically it means convert, succeed in understanding, etc. With the, I think that idea clear, that is converting, deciphering. That was done by Marshall Nirenberg and Heinrich Matthaei. Now comes famous Indian scientist, Hargobind Khorana, but he became American, etc. Argovind Khurana developed the chemical method, was instrumental in synthesizing RNA molecules with a specific combination of bases. And two famous terms in NCERT, homopolymers and copolymers by using synthetic DNA. Polynucleotide with the non-repeating sequence. Typical example, a famous alphabetic code they given, CU, 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 which will produce two amino acids. Amino acid means triplet codon, so CUC, leucine, UCU, serine. That itself is a question. That is concerned with the famous scientist, Argovind Khurana. Now comes the last scientist in this sequence, according to NCERT. I think first page is clear to all of you. We can proceed to second page. Severo Chao. Enzyme involved is polynucleotide phosphorylase. And what is the aim? Help in polymerizing RNA with the defined sequences in A. Next word is important, template independent. This word we are studying in replication of DNA, transcription of RNA, etc. So here it is template independent manner. That means enzymatic synthesis of RNA. I think these four scientists, their work is clear. Direct NCERT match the following. Any theory exam, NEET exam, they can be asked. Direct NCERT. You can also check in NCERT book. So it will be able to follow the right or wrong. All four scientists. Now, finally, a checkerboard in connection with the genetic code was prepared. Checkerboard is an assembly of bases here. First chapter genetics also we studied checkerboard. Punnett score, etc. Here, checkerboard. And this is famous checkerboard with 64 codons. And how we'll write this first? That I will explain first. This is connected with the mRNA. That is why UCAG. So 4 bases, UCAG top, UCAG bottom. Now simple procedure. First, writing 64. No need to by heart. See, first line with the blue color you can find. Write 4U, 4C, 4A, 4G. For that, no need to by heart. Middle line, somewhat red colored arrow. You can find U, 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 U. Here also 4U. Here also 4U. Here also 4U. So second middle line is same letter. 16, same letter in all case. And the last one, the brown line, simply write UCAG, 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 UCAG. Here you can find the second row 16C, 16A, 16G. This way you can easily write, even no need to by heart. So writing over. Now, that is how we can represent it. First, we can discuss with the salient features and remaining things. So writing 64 codons over, no need to by heart it, but many things we have to learn there. That we will be learning. First is triplet code. So for one amino acid, there will be 3 adjacent nitrogen bases, triplet. So example, U, U, U. Here you can find it is for phenylalanine, PHE. Total 64 codons, 8 into 8. So 64 codons, but 3 codons will not code for amino acid, but they have an important function, and they often refers to be nonsense codon or stop codon. We'll be observing it later. Second is start codon. Start codon is AUG, will normally act as start codon. It codes for methionine. Start codon is also called as initiator codon. Third one, you can find this will be mRNA. It will be mRNA. mRNA, the message is read in a continuous, NCERT term is contiguous fashion. No stop, no punctuation, no comma, nothing. Continuous. Next term is stop codon. Everything written, this will be easy for you. Stop codon. Stopping of the elongation of the polypeptide chain is done by stop codon. Normally, 3 stop codons are there. They will be named as UAA, UAG, UGA. You can find. Some other terms will be there like a synonym. UAA, which is colloquial. UAG, that is otherwise called amber. Amber means resin. UGA, it is called opal or diamond. So that 3 terms also important. UAA, UAG, UGA, ochre, amber, opal, respectively. AUG will be normally acting as start codon. That is a general rule. Generally, AUG will be start codon normally and will code for methionine. But there are certain exceptions. GUG can also act as a start codon in bacteria and mitochondria. That way, various variations are there, but that part is not directly coming and said, but just mentioned here. Start to stop codons also called punctuation codons. Stop codon is also called nonsense codon because initially scientists thought there is no function for it, but now we understood it has today important function stopping. I think this page is clear to all of you. 64 codons, how we'll be writing it. Now we can proceed to next area. Yes. Features of genetic code, we are proceeding. Fourth one is degeneracy of genetic code. So what is meant by the term degeneracy? See, normally triplet codon codes for amino acids. There are amino acids coded by different codons or more than one codon for certain amino acid. Then the code is supposed to be degenerate. So different or more codons coding for same amino acid. Code is supposed to be degenerate. In this respect, we can find how from this table questions comes. Typical example, proline, valine, alanine, glycine, threonine. 4 different codons will be there. Everything I marked with the different colors, etc. So you can easily observe it. Okay, so proline, valine, alanine, glycine, threonine, all with the 4 different codons. Serine, leucine, arginine with the 6 codons maximum. Methionine with 1, AUG, tryptophan is 1 with UGG. So methionine and tryptophan degeneracy will not work. So exam point of view, that is important. So how many with the 4, how many with the 6, how many with the 1, degeneracy is absent in. You can, this way, you can write some more amino acid with more than 1 codons, etc. And make a ready reckoner and make a suitable mnemonics because it's an area of question. Typical example, CUU, CUC, CCU will be coding 4. So we'll be able to say it is leucine and proline. Not easy. By practice, we have to develop it, make a mnemonics that is better. Question will be there for theory and NEET. Fifth one, universality. Genetic code is almost universal. A codon will code for same amino acid in different organisms. Same amino acid, different organism. Some exceptions may be there, but generally same amino acid. Next is polarity. See, based on universality, some products are formed from bacteria. I think one example is humulin connected with the bacteria. Next is polarity. Genetic code is always read in a fixed direction, that is usually 5' to 3'. Seventh one, genetic code is normally non-overlapping. Means one codon is independent to other and will not overlap the next codon. Eighth one is non-ambiguous codon, otherwise called unambiguous. Means one codon specifies only one amino acid, will not code any other amino acid. So unambiguous or non-ambiguous. So important area for question, theory, NEET, characters of the codons. So when we say degeneracy, what is the reason for it? Many reasons are there, 2 or 3 we can discuss. See, one reason is it is concerned with or connected with the tRNA. First position of anticodon on tRNA, normally an unusual base, like inosine or dihydrouridine or pseudouridine. That is uniqueness to transfer RNA. This unusual bases at first position of anticodon, base only, first position of the anticodon will base pair with more than one base in the third position of the codon. So inosine can pair with the uracil, cytosine, adenine. Uracil, cytosine, adenine. So that is how that degeneracy develops. One reason. Second one. Say one important enzyme we'll be learning next episode, that is amino acyl transferase synthetase. So some mistakes connected with this enzyme in attachment with the proper amino acids. And third one, number of transfer RNA in relation with the, that is in the next page, I think. Number of transfer RNA in relation with the number of codons. Some reasons why degeneracy arises. Not directly mentioned, but better if you know 1 or 2, it will be good. Next comes it, again, not in NCERT, but we should know it. Wobble position or wobble hypothesis, connected with the famous scientist Francis Crick, 1966. Wobble, what is the meaning of the term wobble? Wobble means change. That is the meaning of the term wobble. Meaning is change or move. In degenerate codon, normally first 2 nitrogen bases will be same, but the third one is different. But the third base has no effect on coding. It is called wobble position. I'll show it. You can find here. See, here you take CU, CU, CU. All are same. Here only difference comes UCAG. So that is called wobble position. I think idea clear to all of you. Okay. Now, Forward question clear. That is concerned with the transcript, not directly mentioned in NCERT. Next is, what is advantage of this idea? There are few advantages for degeneracy to the organism because change of mutation can be reduced. It may cause silent mutation and connected with the efficiency of transfer. In this way, few advantages are there, one or two just to mention. Now we can proceed to mutation and genetic code. Mutation already we studied still. How genes and DNAs are related, easily understood by mutation studies. The effect of deletion or insertion or rearrangement in a segment of DNA can be easily followed by mutations like point mutations, frame shift mutations, etc. Typical example is sickle cell anemia. Already we know what is sickle cell anemia, transition, transfer, always studied. To get it in a more simple manner, few examples from NCERT itself is given there. Page number 97, reprint edition, March 24. Again, direct NCERT theory and question. Now, insertion, a letter of B. Everything you can find there. Here you can find B. In between has an red. So, original statement was RAM has red cap. After has, a B introduced here. So, meaning changes. Now, you can find two letters added as BIR. Then, one more letter. So, like triplet is added. RAM has big red cap. So, except big, if it is not there, remaining four are same. Same with the deletion. We can find RAM has red cap, R gone. Then E gone, so RAM has DCA. Then D gone, RAM has cap. So when we come here, some meaning is there. So this way things goes on. So what is a nutshell or conclusion? Insertion of deletion of one or two bases changes the reading frame. No doubt, from the point of insertion or deletion, from there only it comes. However, such mutations are referred to as frame shift, insertion, or deletion mutation. Insertions or deletions of three or its multiple bases insert or delete in one or multiple codon, hence one or multiple amino acids. The reading frame remains unaltered from that point onwards. That means RAM has big red cap. So after that, again, red cap comes. That way. I think idea clear. That is regarding how mutations and genetic code has been linked. Now we'll be proceeding to last topic of today's discussion, tRNA. Detail manner given there. Transfer RNA, adapter RNA, soluble RNA, sRNA. So tRNA constitute about 15% of total RNA. We already know it. It is the smallest RNA among rRNA and mRNA. Francis Crick postulated the presence of an adapter molecule that would be on one hand, read the code, on the other hand, would bind to the specific amino acids. This adapter nature was established much later, NCERT page number 89, reprint edition 2024 March. Now, models of tRNA. Basically for us, two-dimensional model and three-dimensional model. Two-dimensional model, more important for us, three-dimensional also mentioned. So diagrams you can find. Two-dimensional model, it is clover leaf-shaped model. This is three-dimensional inverted L shape. So all details are given there. This is NCERT. These all are NCERT, but presently not there. Still, I put all this together, so it will be interesting to watch it. Now, normally, all tRNAs got almost similar structure. Number of types of tRNA, number of types, not number of tRNA, types of tRNA varies from organism to organism. Generally, more than 60, because 64 codons will be there. The theoretical maximum possible, that is more than 60. May come below 64, that way. Because that is why the value given more than 60. I think idea clear. So this is two-dimensional clover leaf model. Scientist is Robert W. Hawley, 1965. And this is inverted L shape given by Kim et al. 1973. I think that idea is clear. Now we'll be proceeding to further detailed study. So we can proceed. Diagrams, this is two-dimensional, this one, and this is three-dimensional. So when we start with the two-dimensional, it's better. From here, 5' and we can start. So 5' and when we start, there is phosphate P, then G. After that, it comes down, enters into first loop. So from 5' first loop. Then goes down, second loop, then extra arm, third loop, then fourth loop, that is first from third dash or 3 dash. I think that arms are now clear. 1, 2, small arm, 3, 4. Now, here you can find anticodon that pairs with the codon. This all will explain. First, simply explaining the loops, all these things, direction, etc. And this arrangement, take care. 5 dash, P, G, loop 1, loop 2, loop 3, loop 4, then C, C, A, OH. 3 dash and we are reaching. So this is an important question for exam, sequential order. Now, 3 main loops in transfer. Main loops in transfer includes amino acyl synthetase or DHU loop. Here you can find D loop, dihydroxyuridine. D loop, that is first one from 5'N. Direction we should take care from which side, 5' or 3. Here it is from 5'N. Ribosomal binding loop with the 7 unpaired bases. That is first from 3' direction here. It is size C loop. This is DHU arm, D loop, T arm, etc. Also synonymous. Now comes anticodon loop with the 7 unpaired bases, of which 3 bases act as anticodon for particular triplet codon present in mRNA. So this will be acting, pairing with the codon. The unpaired base, 3rd, 4th, and 5th from here is the in the anticodon loop of transferrin pairs with the codon in mRNA. So 7 unpaired bases here. 3 bases act as really anticodon for a particular triplet codon present in mRNA. The unpaired bases will be 3rd, 4th, and 5th in the anticodon loop of a tRNA pairs with the codon in mRNA. This is 3-dimensional diagram. Everything marked there, so it will be easy for you. Most among these two, this one is more important. 2-dimensional view, clover leaf shape model. I think that is clear. Now, 5 theory question, 5 NEET model question with the key given here. So what all things we learned in this episode? We can just rewind it. So we started from, or we just having an introduction, then genetic code, then transfer RNA, 5 theory question, 5 NEET model question with the key. Hoping that it is clear to all of you. If you like this episode, subscribe or like it, share it, comment on it. We can meet in the next episode. So take care. Bye. Thank you.